Thank you, Hirokazu, for such a wonderful and beautiful Kyudo ceremony. Good evening, my name is Antoine Scott, and I'm the co-curator of the forthcoming exhibition, Emergence, a Genealogy, which seeks to explore the complex yet fascinating community of bioart and artists that intersect with the emerging and understandings of synthetic biology, its tools, and its practices. Emergence is currently one of dozens of exhibitions currently being supported by the Pacific Standard Time initiative of the Getty. I'm pleased to introduce this evening's event, 1000 Days, which serves as a programmatic expression of ongoing research for our exhibition. If you have not done so already, you can find out more information on our website, fathomers.org, and by signing up for our newsletter. You can also find us on social media at Fathomers. Before we begin, I'd like to acknowledge those who have made 1000 Days possible. The Getty Foundation as a part of Pacific Standard Time, Pat Wyatt and Hirokazu Kosaka here at the Japanese American Cultural and Community Center, Sarah Connolly Odenkirk, and our board of directors and Fathomers family and friends that are in the audience with us this evening. Thank you for your continued support. And now to our program. This evening we are joined by three individuals whose professions and passions lend themselves to answer some of the outstanding questions that still remains as a part of our research for Emergence, a Genealogy. I'm so excited to have the opportunity to learn from them with you tonight. Our moderator is Dr. Yewande Pearse. She is a trained neuroscientist, stem cell biologist, and science communicator who has dedicated over 10 years of research to understanding rare genetic brain disorders at King's College London, the Institute of Psychiatry, Psychology, and Neuroscience at the Lundquist Institute at Harbor, UCLA. And she is currently working in the world of biotech and is also the host of the podcast, Inside Biotech, presented by Biotech Connection Los Angeles. Her communication work exists at the intersection of art and science and culture, where she strives to make science accessible for all. She has produced and hosted the Sound Science podcast by Dub Lab Radio and has collaborated with Headspace, Natural History Museum, Female Collective, Seed, Science Gallery Detroit, Naval, Massive Science, and TED Med. Dr. Pearse is joined this evening by Dr. Drew Endy, who is a member of the bioengineering faculty at Stanford University and BioBricks Foundation president. His research has pioneered amplifying genetic logic, rewritable DNA storage, reliably reusable standard biological parts, and genome refactoring. Drew helped launch the new undergraduate majors in bioengineering at both MIT and Stanford. He has also co-founded the iGEM competition, a global genetic engineering Olympics, now with over 6,000 students participating annually. In 2013, Drew was recognized by the White House for his work on open source biotechnology, and more recently, he received an honorary doctorate from the Delft University of Technology. Drew has served on the U.S. National Science Advisory Board of Biosecurity and the Committee on Science and Technology. He currently serves on the World Health Organization's Smallpox Advisory Committee, as well as being recognized as one of the 21st century's most influential people by Esquire magazine. And joining the conversation is Dr. Jennifer Willet, who is the director of the Incubator Art Lab and Canada Research Chair of Art, Science, and Ecology at the University of Windsor. In 2017, Willet was inducted into the College of New Scholars, Artists, and Scientists of the Royal Society of Canada. In 2009, she completed her PhD in the Interdisciplinary Humanities program at Concordia University and opened Incubator, the first biological art lab in Canada. Jennifer is internationally recognized as an artist and curator in the emerging field of bioart. Her research resides at the intersection of art and science that explores notions of representation, the body, ecologies, and interspecies interrelations in the biotechnological field. She engages in performance, installation, photography, sculpture-based practices, as well as community arts, social practice, philosophy of science, media studies, science and technology studies, combined with protocols and life forms from the biological sciences. So with that being said, panelists, may you please take your seats.
Thank you, Antoine. That was a really nice introduction. I'm really excited to be having this conversation and meeting you in person, Drew and Jennifer. We have an hour for this discussion. I feel like this discussion could be as vast as synthetic biology itself. So let's jump straight in. So on the topic of the vastness of synthetic biology, I think during this conversation, we're going to start we're going to zoom in, and then we're going to zoom out, and then maybe we'll zoom in again. So I thought it would be fun just to start by thinking of 100 days and what that concept is. And Drew, a lot of your work actually is centered around this concept. So with biotechnology, I think it can be very vague. And as the general public, we see the end products, and it's really amazing and wow, but actually the intricacies of what goes on behind that and the prelude to that is always very mysterious. So I was wondering if you could demystify some of the work that led to Pure, which is an artificial model system with the potential to remake 36 life essential functions and the prologue to the 1,000 days concept.
I'll try.
In about 5 minutes or so.
Thanks so much, and incredibly grateful to be here in conversation with everyone. Synthesis, I actually kind of want to just attempt this by starting with the word synthesis. And what does that mean in its root? It's a putting together, composition. And we're familiar with synthesis in the world of art and in music and in everything in which we have agency. For the history of life synthesizes itself, but our relationship to it is it happens to us. And so when we take this perspective of intentionality and the responsibility that comes along with it, and we're putting together life, is it sublime? Is it beautiful? Is it scary? It's probably mostly scary or just doesn't work, but sometimes it's sublime and beautiful. And so in the root of what's happening, I hope you might see everyone a connection between the making of things, whether it be art or living systems. Since you asked specifically about Pure,
yes,
in this environment, I want to acknowledge where Pure comes from. So this is a research innovation about 20 years old from colleagues in Japan. And it stands for protein production using recombinant elements.
Let's break that down.
Okay. What they figured out how to do was purify from existing life forms all the biomolecules needed to, when you mix those molecules back up in a tube and then add whatever DNA you want to the tube, the message encoded in that DNA will be made by the Pure system. And it's called Pure because they figured out exactly what molecules are needed to do that bottom-up reconstitution of in a tube you could mix up from scratch, take any DNA and have that DNA make your favorite molecule. And so just want to acknowledge that's an innovation established out of Japan 20 years ago, and is the starting point for what we're attempting over the next 1,000 days by asking the question, if you could Pure make Pure? Could Pure remake itself? If we added the DNA encoding the molecules that make Pure to the tube, do we get more of that? And that would be the beginning of an autocatalytic cycle.
So it would be a replication process.
That's right. A growth process.
A growth process.
A synthetic growth process for which every component is known to us. We might not perfectly understand everything, but at least we know who we're inviting to the party. Yeah.
That's an incredible concept. So how close, I read the paper and I know that it was 36 molecules identified, so it hasn't quite worked. So the limitations in that.
Let me describe that and then offer some quick context. So, can pure remake pure? No. That's the answer. And there's two challenges. The first is, like, how much oomph does it have? Every time you make a molecule, you have to spend some resources or energy, some juice. And the mixture that you could, by the way, anybody here could buy a little container of pure. You can buy it from New England Biolabs if you want, neb.com. No affiliation, no endorsement, amazing, right? But this tube of pure doesn't have enough oomph. It needs like 20 times more power or juice to be able to remake itself. But we think we can solve that problem just by adding in more energy. But the second challenge, technical challenge, and this is, I can't believe we're going here so fast. This is like frontier esoteric. Let me see if I can do it. Remember that the molecules that make pure are purified from existing life forms, which means that their making inherits all the capacity and context of the life forms they're purified from. If we put the DNA to make those molecules into a mixture that only has those molecules, we're missing the much bigger, complicated natural context that might derivatize or subtly change in some ways we know or some ways we don't know, mysteriously, the molecules such that they function differently. So the technical innovation we've realized is we've learned how to ask and answer the question, can pure make itself? And we know the answer is no, but now we also know how to debug, like why not? And what do we need to add in addition to the mixture to make it turn over? And so that's why we talk about it in the context of 1,000 days. We started this work 6, 7, 8 years ago, and it's just total ambiguity. Like we don't know how to go about doing this, and it's the creative work of the graduate students, the PhD students, and their superpowers of navigating this ambiguity, who have gotten us now to what we think of as a starting line. Like we know how to coordinate the work, we know how to debug the puzzles, and so we're ready to go. And when we think about how much work is ahead of us, feels like it's about 1,000 days of work. What's the finish line look like? You could mix molecules together in a tube, derived from many different natural life forms, but separated and recombined like Humpty Dumpty, like putting it back together, and it works. So like we're trying to solve the Humpty Dumpty problem is another way we think. But anyway, that just feels like, for reasons we might explore, I don't know, like that feels like an important spot to get.
I mean, I think that's an incredible way to start just because of that detail and the fact that it is a very viable possibility, and now we kind of have an idea of why that is. And so thank you for sharing some of that detail. So I said we'd go in and then we're going to zoom out. So Jennifer, your work as a bioartist and director of the Incubator Hybrid Laboratory, it challenges synthetic biology and its application. So we've just heard from Drew a very specific application of synthetic biology, and it will shape our conversation today. But thinking about 1,000 days, and I know that you've read the paper, where do you think art is most pertinent in this concept? Because this is an incredible technological development. So in terms of bringing those two worlds together, where do you see art?
I'm not sure I can answer this larger question, like what is the role of art in Drew's project, but I think I can answer a very specific response to that question, is that why might I be here to discuss this project with Drew? So I'm a bioartist. I work in art and biotechnology. I have no scientific training. I read a lot of scientific papers, and I sort of like adopt scientists, and I get them to teach me what they know how to do. And so for many years, I would just travel the world, and I would go into labs, and I would try what they did, and then I would make some art projects out of that. And I found that I was kind of a disruptor, but also a friend and a provocateur. And often at the end of those residencies, the scientists would say to me, you know, I got a lot more out of that than I thought I would. I learned something new about my practice. So about 10 years ago, I decided to build my own lab. I was tired of traveling. And I built an art and biotechnology lab at the University of Windsor. 10 years in now, or 15 years in, we have 2 facilities. One is a BSL Level 2 lab that is also a performing arts venue that has theatrical lighting, sound, those types of things. And the other is a BSL Level 1 lab where we do community engagement workshops. So the reason why I think I'm here, and maybe what I have to offer, what you're talking about, is that over the years, science itself has become my topic of interrogation, and not just big capital S science, but all the little minutiae, like the bureaucratic processes, the hiring processes, the methodologies that go into making science. So for example, I'm really interested in moments of biosafety certification. Like at what moment does a room no longer become a room, but it's a lab, and what happens in that space at that time? How can we push the limits of our relationship to that transformation? So I suspect I'm here to sort of talk maybe with you a little bit about some of the processes that are involved in these large collaborative projects that seem to involve all different types of scientists, but maybe also thinkers of different kinds. The 1,000 Days prospect is also like an interesting short-term turnaround reality TV maybe proposition. Like, you know, people like, what can I do in a year? Or so I'm just here to really have that conversation with you. I'd love to learn more and ask questions more about how your lab and this project, because it's not just your lab, functions.
Yeah, thank you. That's a great answer. And I think just that comment about how you can actually acquire this vial of pure, it's available. That distance between, okay, it's
fascinating what you can order online, or like 1-800 Lab Rats, for example. The shipping and receiving office at my university is terrified when packages come in with my name on them. So I will probably order some pure after this discussion.
So I think on the topic of 1,000 days and whether we could actually create life in 1,000 days, I guess the first question to really dig into and maybe unpack is why we would want to do that. And so from different perspectives, what does this really say about us? What does it say about synthetic biology? What does it say about art? So the idea of 1,000 days, it's ambitious, it's impressive, it's also audacious. So reflecting on the needs of humanity, what is the goal, would you say, for actually creating a cell from scratch? Like, it mirrors a lot of the beauty of life and the creation of life. So pure making pure, for example, or the wider question, what do you think humanity can do with that?
Yeah, I can certainly offer an argument or perspective from utility, but I think that I want to offer a different one. So I wish for you to imagine what it's like to live in the bottom of a well, a burbling basin of water flowing up from the ground. I wish for you to imagine living in the bottom of a well, a life well. We live in a life well, in a basin of attraction which holds the forms of life that we are and that we're familiar with. And we're in the bottom of the well because we're trapped. We're constrained. We're constrained by lineage, our parents, the life that came before us. We're constrained by the requirement of, if life's going to continue, it has to reproduce. We're thirdly constrained by, if the environment is changing in our well, life better be able to change too. It better be able to evolve. And so being within a well is wondrous and sacred and safe and protected, but it's also like we're trapped down here. And I want to clarify a thing. We don't use the word create life. We use the word construct. And that might sound pedantic, but we mean it. You know, like I grew up my summer internship was for Amtrak doing construction management, like structural engineering and excavation. And I'm familiar with the laboring of an engineer, even though it sounds like I'm doing science. I'm like bio bricks, right, and mortar. And so it's like, I've got a budget. I don't understand exactly what I'm doing. I don't have perfect capacity to shape reality. I'm this omnipotent chemist, So anyway, like, we're in this life well. And the possibility of constructing life is like climbing and clawing and scooching up to the top of the well, to the lip of the well. And now what's up there? I don't know. But what I do know is possibility is up there, because it lifts us up out of the constraints, these three constraints of lineage, requirement of reproduce, and requirement of evolve. And it reminds me of what I heard about the 1960s. There was another well that we climbed up out of for the first time, the gravity well of the Earth. And with the rocket programs, we launched somebody up into orbit. And of course, what's up there is the vacuum of space and some other planets. It's like amazing, but it's not that interesting. But like the life well, the life well is way more interesting, because most of the possibilities of life have never been explored or even imagined or realized. And so it could be an Eden, or it could be scary. I mean, anyway. So like, maybe that's why we're doing it. It's like we're in this well, and I want to get up there and look around, and I give you the utilitarian dream, but I don't like that. That's like bonus.
Yeah, no, I was actually also going to use the space
Okay.
example. I just in agreement with you, because I think humans just have this natural tendency to want to innovate and explore and find out, what we can do, and it opens up this possibility of things that we don't have the answers to yet. So, Jennifer, I think from your perspective, actually, as a bio artist, your interest in a concept like this, how would you approach it? What would your perspective be?
I have so many questions. Your metaphor of a well, it feels a little religious. It feels like this really progressive, reaching towards transcendent goal. And I think that could be like a useful or motivational metaphor, but I'm not sure it's accurate. So I'm still just reveling in this story of us in a biological well, aiming for the future. I have a little bit of a different take on it. But it's also a metaphor, right? So just understanding that all of these metaphors have challenges or sort of cultural implications. I get a lot of questions from people who are asking me about whether biotechnology is incredibly unnatural, or and then the implication of that is like evil or harmful, and all of those things. And I often argue that actually it's quite the opposite. It's incredibly natural. If you look from an ecological standpoint, organisms sort of consume resources, they excrete resources, they produce waste. Some other organism eats the waste, right? And then through doing that, they build domiciles for themselves, or they gain more energy, or they transform in different ways. And so I actually, when I hear this story, I don't think that it's unnatural, and I love your description of your engineering description of constructing versus sort of like a Genesis story, right? So you are constructing these molecules together. But I think that if you take into its logical extent, what we're doing in the lab today is natural and is just like this ecological thing that organisms tend to do, is to consume all of the resources around them until there's nothing left, and then they die. And so that's sort of the way that I read what you're saying, is that that we are doing in the lab, that we're exploring the parameters of what is possible in the lab, And one of the sort of ultimate goals might be making new life rather than reproducing existing life.
No, I think
Yeah,
these are
I
two
wanna-
very
let's
good
go.
perspectives. So I
Can
want
I
to hear
respond?
what Drew has to say about that.
So I have to go to moral compass. Like, what's my moral compass? And if you don't like my moral compass, you got a better one, you can upgrade mine later. Like, I'll take yours. Not steal it, I'll just like adopt it. So my moral compass is am I contributing to improbability? By my decision, am I making things more improbable? And so how does this relate to what, so the way I think about it is life exists on a gradient of energy, mostly from stars to the heat death. And on that gradient, there's a metaphor, a water wheel. Living systems harness some of that energy, and they organize that energy into relationships among molecules, among cells, among organs and organisms and ecosystems. And we get these improbable living patterns that promulgate through space-time. And that's what life is, so far as I can understand it. And so if by my action I am contributing to these improbable living patterns, then directly or indirectly I am contributing to the good. And if I am destroying or reducing this improbability, then that's a bad thing to do. And so by this frame, I find myself through my actions, I don't see any distinction between human or nature at all. I'm just part of life, and I am realizing and contributing to life by this type of action. And getting to the capacity of being able to construct life from scratch without these constraints is like the ultimate platform or starting point for producing improbability. And so I'm attracted to it because it feels like the fundamental good that is at the core of my moral compass. Does it mean that everything that will happen with this will be? No way, right? And I'd love to be able to like talk about the fear and scariness and terror of it, but anyway, like we're on this and anyway, like we're on the improbability tour is what we're trying to do.
I'm feeling 95% of what you're saying.
What's the 5%?
Yeah, let's talk about the 5%.
The 5% is the part where you say, and that's good.
Yeah.
Right? Like contributing to biodiversity, and I describe it, I'm interested in contributing or being part of the unfurling of the universe, which I think is a very similar sensation that you're talking about. There's like an entropy, and the unfurling of the universe for me is neither good nor bad, and the outcomes are not good or bad, but I enjoy the sensation of participating in that unfurling. It's just the part where you say, and that's good. But that's okay. We can be different.
So we don't know if it's good, and that's the point of contention, because it's all about relationship. It sounds like we can have this, we can create these synthetic biological systems that fit into our living systems as long as it's all in harmony, then that would be good. But there's the improbability and unpredictability.
Harmony is impossible. That's not going to happen either, right? Like if everything goes well, there's always going to be challenges or suffering and the end of life, right? That's also part of the natural world. I think I'm just really puzzled that I'm sitting next to a world-renowned scientist who is emphasizing like this goodness when I would expect that from me as the artist. So it's just a really interesting inversion of the descriptions of our disciplines.
Yeah, well, I see a lot of overlap between both of your worlds and perspectives, and I'm excited to continue this conversation. Just thinking about this idea of construction and building and the 5% of, that's good. So what needs to be built in when we're building to kind of control for some of that? Drew talked about fear, so people do have a knee-jerk reaction because of the unknown. So what should we be building in? Or should we? Or should it just be, let's see.
So that's a great question. We don't know. And so we're figuring it out as we go along, and we would take upgrades happily. we learn as much as we can from what we can study. So the Apollo program that got a person to the moon and back a couple times, that's interesting, but note that it didn't routinize bringing people to the moon and back. It was a moonshot project and no infrastructure built along with it, right? Like we just did it and then we stopped doing it. So we look at that and we say, that's not what we want to do. We want to, we use the word routinize the building of cells. That might not be the right word, but that's one choice we're making. And so that changes how we go about the work. We might not be the first to do it, but we're doing it in a way that allows us to continue to do it on a sustained basis. Another thing that's a choice we've made is to, you could go after this and said, I've done it. Ha ha ha. Not you. Right, good luck catching up.
Competition.
Right, like good luck. Or you could say, well, no, let's if we're climb up the well together, leave some handholds. Sorry for the I'm going there, right? And so in taking inspiration from the architecture of the Unix operating system that underlies the Linux open source operating system, like literally tracking down the videos of Dennis Ritchie and colleagues in New Jersey in the 1970s and the cultural decisions they made about how to architect that technology, about enabling a fellowship of programmers. Literally what they say, right? We're very inspired by that. And so we've adopted the phrase, like how we organize ourselves will impact how we organize ourselves and vice versa. And so we're trying to build architecture that enables community, even though the community doesn't exist in this way right now. Like there's a beautiful academic community working to build cells. The Dutch, the Germans, the Japanese, the Americans, all over, like beautiful academic community. But academics are kind of like cats. Like we're rewarded for being idiosyncratic and different, right? More often, and so I don't think that anyway. But we're making these types of choices, and again, like you can program us, like if you would rather like tell us the other thing to do. Like, okay, we'll try it out.
So I have some suggestions.
Yeah, I was going to ask Jennifer. I'm sure you have some suggestions.
So in my lab, I now run my art practice the way most scientists run a lab.
Do you call it a lab or a studio?
I call it a lab.
A lab, okay.
Yes. So I have a laboratory research group, and we have team members from all different backgrounds who are working on my projects right now as we speak.
Okay.
We've come up with some ideas about how that might work. We've been thinking around the idea that biotechnology might be a technology of love. And what would it be to practice interspecies relationships in the lab with the notion of love as a motivational factor? I would like to first say that I do not mean romantic love or Hollywood love. I mean bell hooks, committed political durational commitment and inter-exchange love. So there's not a lot of hand-holding and that type of thing in my lab. And then it grew, and I got thinking about not just the relationships we have with the organisms, but the relationships that we have with each other, right? Who is allowed in that lab? What backgrounds do they come from? What knowledge are they bringing to the table? So I have artists and scientists, but I have philosophers and I have nurses. I have people from very diverse cultural backgrounds all working in my lab. The diverse age range in my lab is from 19 to 69, I want to say. And what happens is that then we have this chorus, this cacophony of interrogation around ideas. Now I'll say one of the drawbacks of that is 1,000 days, it's slow. We got to talk about each other's feelings. We need to learn about each other's backgrounds. It's a little more, you're not going to do anything in 1,000 days in my lab. But the outcomes and the community that we build in that space are really profound. So when I read about your work in this paper and listening to you now, I'm thinking a lot about the pandemic And I'm thinking a lot about how in less than 1,000 days, we came up with a vaccine internationally. And on the one hand, for me, this is like one of the epitome examples of what science can do when we break down international borders, boundaries between corporations and governments and these types of things. But also, the rollout of that vaccine made visible to us the areas where we are failing, right? So there were all sorts of communities who did not feel a trusting, loving relationship with those vaccines and were unwilling to try them. And so I think that what we can learn from that and what might be really valuable for your project would be to widen the scope of who is involved in the production of this research and what are the discussions. Not just a technical discussion of how to build this cell, but the cultural discussion at the front end of the production of the cell. Thinking about what communities are going to have access to this cell, how will it benefit them, how would they like it to benefit them instead of how you think it might benefit them. I think that there's some real opportunity here to learn what we saw as the successes and failures of the pandemic and how that could transform scientific methodologies to be more inclusive and more polyphonic in the number of voices that are involved.
Yeah, thank you, Jennifer. I
Amen.
mean, before Drew responds to that, I think this is really one of the reasons why I was excited for us to be on this panel together, because I begin to see where this overlap could happen. So I think just thinking and spending a little bit more time on how synthetic biology could become more collaborative and impactful to, everyone. Drew, in the context, like if we zoom out and take 1,000 days and just put it in the context of synthetic biology, so at Stanford, you actually do have a specific approach to synthetic biology where you're using PACE layers, which is about thinking how to foster a more inclusive synthetic biology culture. So I'm just wondering how that overlaps with some of the things that Jennifer just uplifted now.
I think those are 100% amazing suggestions and beautiful, and more of that's needed. I was teaching last year, and it was an introductory course, and we were teaching in the context of a pandemic on topics of bioerror, lab accidents, or worse, bioterror, somebody causing harm. And a colleague in art practice, Paul D. Marinas, was auditing the course, and he said, "This is a very good week, but you've left out one of the ers." I said, "Well, which one?" And he says, bioscarer. And I'd never heard this word before. I go, "What does that mean?" And he says, this synthetic stuff, it's just scary, man. I'm like, "What do you mean?" We're just like nerding out because it's scary. And to your point, it's slow going, right, to have dialogue across perspective and discipline and background and dreams. And so it took me quite a long time to appreciate what he meant. And what clicked it in for me was when I realized what I'm afraid of. Like, I'm afraid of heights. And my wife, she's really afraid of spiders. And when he was saying bioscarer, that's kind of what he meant. That just like this biotech stuff can be scary. Mm-hmm. And like where that fear comes from might be similar to where the fear of heights comes from, which I actually don't really know where that fear comes from for me, but like that's true. So that lesson from an art practice colleague, Paul, has been so profound for me because it resets dysfunctional relationships. If I were to note that even with privilege, we still have a culture where it's kind of okay to admit that I'm afraid of heights, right? But if I were to go on Twitter tonight and say, I'm afraid of GMOs, what's going to happen is Nobel laureates are going to start tweeting back saying that I'm committing a crime against humanity because this is useful technology that's saving lives. I can give you the letters they write that say this, right? And so like we've got, in the absence of this profound laboring to relate and to connect, we've got these broken, I don't even want to call them dialogues, but you tell me what to give this, and this gift of recognizing that that certain profound elements of this are just scary, is okay. Well, let's talk about that. Let's at least acknowledge it's existing, and then maybe we could learn to become courageous operationally together. So I just want to return to what you were saying, and it really lands with me, and I respect it, and I'm a student of it.
Mm-hmm.
You know what I mean?
I see some of it in your writing, that
yeah.
you're thinking about these questions, about how to
Yeah.
engage with other communities.
Yeah.
Yeah, I think going back to that quote about the history of science and technology teaches us how we organize ourselves will impact how we organize ourselves and vice versa. And I think, Jennifer, so much of what you said just about the trust and the parallel to the vaccine and the pandemic, and especially about this urgency around 1,000 days and that mentality to just move forward, but actually thinking and slowing things down so that you can create room for all of those sort of more nuanced questions, I think is a really, really important thing. So, I mean, scientists are taught that science is objective, and it's not. I don't think we're free from bias, and our ideas are only going to be as good as the assumptions of the people involved. And you mentioned inclusivity and having a lot of diversity. So I wondered if you could sort of maybe comment on how we could redefine those disciplines and individuals that are included. So, Drew, in your lab and the project that you did with Pure, where you're inviting people to partake in that, how does this actually marry up in terms of bringing those two things together? Does it need to come from the individual, like yourself, Janet? I mean, you created a whole lab so you could do this work, and you have people in your lab. Drew, the people who work in your lab, how does that actually translate?
At the end of the day, with like a lab is a relatively tiny place, and as much diversity as you might bring into it, it's always going to be selective. I would respond by sharing the following set of questions. How many, and they're questions related to optionality. How many people should have the option of learning to read and write? Basic primary education. Apparently the United Nations says that's a human right, so we think it's everybody.
Yeah.
And then well, how come? And it's related to your question, right? So that everybody has that optionality. How many people should have the option of learning to read and write a computer language like Python or HTML? And apparently we're trending towards everybody, because we have things like computer science for all in the US. Like let's give everybody the option of learning to become literate with respect to expressing intentions with electrical computing machine. So then the third question and last one in the series is, like, how many people should have the option of learning to read and write DNA? I'm going to say everybody. All right, and I practice that behavior within a context that's not the starting point. The starting point of the practice of reading and writing DNA goes back to, in my amateur understanding, the fear of being bad parents, captured in one form by Mary Shelley with Frankenstein, that inevitably as parents we will fail our creations or constructions sometimes, and that fear is so profound. When genetic engineering got invented 50 years ago, it came into the world with that current of fear carrying it along, and the nerds, all they could respond with was utility. We're going to cure disease, right? That's a good thing to do, right? But it just tried to counterbalance or push back fear with utility without ever really resolving the fear. So like, I've been called the scariest person somebody's ever met, right? When I start talking in rooms where people are worried about bad actors, right? But it's because so there's this coupling between fear and diversity and accessibility and optionality and citizenship are people going to be citizens of the biofuture, or are they just going to be patients and consumers or subjects? Or like, I prefer citizens. But I want to acknowledge, like, we are operating in an inherited context. Never mind the broader structural challenges and inequities, but particularly within the life sciences and its culture of practice, there's a whole bunch of lock it up, keep it hidden, stay away. And there's valid reasons associated for that, but it's the opposite of where I'm trying to get to.
Yeah, absolutely. I think there's a space in between the two where there are people wanting to push the needle, and there are both sides of the extreme. And I think the fact that we can have conversations like this, I actually can't believe that time's actually pretty much up. I could have this conversation for hours, but I think that's really where the work is being done, and where I see 1,000 Days Beyond that pushing synthetic biology and its application forward. So I think we're at time and just looking for a signal. Yeah. So I think we're going to wrap up, but Jennifer, I just wanted to give you an opportunity just to respond before we end.
Sure. I think we really align in a lot of different ways, but I'm really, the thing that I offer and that my community can offer to your community is that it's not just that the people who read and write DNA should be in the room who determine how to read and write DNA going forward. You want people in the room who do not know how to do that, but have other knowledges, experiences, goals to offer your project. And we were talking beforehand, I read a lot of scientific papers, and sometimes I understand them, but sometimes I don't. I read them like concrete poetry. It just sounds like sounds. But there's something about having those other communities and those other people in the room that might not only transform for the better, for ethics or those types of things, but also improve the research that happens in scientific labs. And that is what a lab like mine is trying to do.
Amazing.
This has been a beautiful conversation. I love the tensions and the overlap, and I hope everyone has enjoyed that discussion too. Thank you so much, Drew, and thank you, Jennifer, for being our panelists this evening.
Thank you.
Oh, we actually have time for a couple of questions from the audience. Does anyone have a question for Drew or Jennifer? At the back, we have a microphone. I don't know if you can project, but I can grab it. Okay.
I work particularly with microbiologists. You don't hear? No.
Closer.
Just
Just
yeah.
hold it closer. You have to hold it. You have to hold it really close.
Can you hear it now?
A little bit. Even
Okay.
closer.
Hello, hello, hello.
Oh, there we go.
There we go. Okay,
Success.
sorry. So I'm an artist. I've been working with microbiologists quite a bit, and some who locate really never seen except by them so far, and they want more people to see them, microbes, a mile under the earth in some nook and cranny. And then, frustratingly, they have to bring them back to the lab, and of course, it's nothing like that microbe will not continue in any way, shape, or form, right, as how it did in its own context. So, and you alluded to that in the beginning. So you were talking about, Jennifer, the cultural context, and that's and everything that could bring to it. But what about the interspecies, quantum? I don't know, everything that's going on in a ecological context. Sorry. How can you can you pull that in, or how are you thinking about that? I guess.
Are you asking about the laboratory ecology? Because
yes.
I would argue that a laboratory has a very specific ecological environment that it's producing. It's connected to our outdoor ecology, but it transforms the organisms that you bring into it, right? So the organism that you find 1,000 miles under the earth, if you bring it into the lab, it's not going to function the same way. So it's a different environment. There's a huge argument in the sciences between ecological sciences and lab-based researchers. The lab researchers often argue that you control all of your variables, so you can produce real data. But then the ecologists would say that the data is never real because nothing happens inside of a vacuum. So to study the same organism in those two different environments produces very different results.
How might that apply to this life in this pure life, which I'm picturing in a test tube or in a petri. It's not in a multi-species, rich context.
I don't know. The two comments I would offer are, in rough numbers, we've only ever observed or described about what's estimated to be 10% of life on this planet, like not even close to most, and in part because of the different environment. It's a type of diversity. I'm so interested in your question in the context of the comments about diversity and sort of who has and what has agency in these. Like, what happens if, like, could I bring a banana slug into my lab? What would it say about how to build a cell? Like, what do the banana slugs want? And so I was just wondering about that. Then the second thing to offer in response is, if you were to come visit our laboratories, they reflect a tradition of sterility and isolation and decoupling and stay away. And like, we just spent a lot of effort to build like a big one of those. And it's like so lonely. It's so powerful. It's like so we're dreaming about a sister building for bioengineering and others, which is we call the biodome. You know, like, instead of 2 go in and 1 come out, like Thunderdome, 1 go in and more comes out, you know? and it's an apex ecosystem that, and the first thing you walk into, and what you do is you listen. And then, like, maybe later it's like dirty and weird, you know, but and then from the engineer's perspective, it's like the laboratory by which we learn how to design and build ecologies. We don't have that space right now, and the point about would an ecologist ever believe this was relevant to the real, right? I mean, that's crazy, but it's really interesting.
Yeah.
It's a really interesting, different perspective.
My lab, we're intentionally not perfectly sterile, and I hire scientists, and it blows their minds.
Yeah.
And
I love it.
so
I love that idea.
one of the goals, I think, in the lab is that I'm training the next generation of scientists to go and build their own labs under these different auspices.
Yeah.
Yeah, that's so interesting. So, as a stem cell biologist,
Yeah,
it's
it's
like
terrifying.
my nightmare is a 30-day long protocol where everything dies because it's not sterile. So
Contamination.
And I'm like, wow,
contamination.
that's the cycle of life.
Or when things grow actually in cells, that's another twist that you try and avoid. But that's a great question. Thank you. We have time for one more quote right here.
In that vein, when you started speaking, Drew, I
Got to
thought
hold it a bit close.
in that vein, when you started speaking about sort of pure 1.0 that didn't quite work, I was thinking it was pure to pure. Like,
Peer to peer.
pure was too pure, right?
Yeah,
It was
yeah.
too isolated. And so, and also going along, like, nothing's in a vacuum. So
I have to be honest with you, even the name pure for me really has like eugenic implications that makes me very uncomfortable. So yes, I would argue that pure might be too pure, but maybe you have to try it first in the lab, and then like maybe the next step is that it goes in the world or interacts with other stimuli.
Agree. We didn't name it. It was named in Japan 20 years ago.
But the ethos of it,
But I
right?
totally agree. We skipped over a little bit of context. And so I want to offer the competing or complementary approach that's really important for understanding the pure-based approach. So there's another approach, which is to take an existing organism and attempt to, in an operational sense, fully understand it. And so you take a life form, maybe it's a simple existing microbe, and you start to remove the things that are not essential. And you keep trying to remove things until you're left with the minimal set of elements or genes. And this work is beautiful science and results in organisms that have, say, 473 genes. Each of the 473 are essential. Now, when you do that, you end up with a puzzle, which is we don't know what all the genes do. in 2016, for example, the last time this was done really well, there were, of the 473 individually essential genes, there were 142 that nobody on Earth understands what they do. Now, that might not bother you or not, that's your choice, but it's like a profound fundamental scientific mystery. It implies that there are physical functions essential for life. We don't know what they are. And so that mystery has been reduced. Now the number is 92, but it's not zero. And so this is the context of that motivates my students, right? It's like my students adopted this bottom-up approach that other colleagues, it was like they had to convince me to go the bottom-up way. And so you're right to point out the peril and limits of pure, right? But the thing that's motivating it is the persistent ambiguity of the things we can't understand. And it suggests an obvious pitfall right ahead of us. If when you whittle down a genome and you're left with the essential stuff, and you have the mystery of the essential genes of unknown function, you can easily expect that the pure bottom-up approach is going to get to a point where we've added everything we can think of that should be totally sufficient, and it doesn't work. The essential functions that remain unknown. And so you like there's a collision course here maybe, or something, or like a duet. I don't know what. But we agree that the name is fraught, and no, it's like, but it's just like it's the, by the way, it's the cultural lineage that we inherit that's constraining us, just to be Captain Obvious, right? And but we're aware that we might get 1,000 days out, and there's this foreseeable mystery, and that's, you could say that's going to be a bummer or really interesting, but we have to at least get there. I'm not going to say that will be good.
And maybe that's why it's never going to be life, or like you said, construct rather than create, because that mystery is just integral to life, and there's always going to be mystery that you can't, use technology to.
The physicists are still studying gravity, but the Golden Gate Bridge is operationally working for now, right? So I think that's how, like, if we ever pull this off, it'll be like the GGB, and the mysteries of life will be explored forever. Yeah.
And that's a great way to end the conversation. Thank you for those questions, and thank you for this conversation.
Thank you. Oh my gosh. Amazing.
I want to talk so much more over dinner.
Let's go. Amazing.
Yeah.
And off we go. Stage left or right. Thank you.